Hello, welcome to Animal Protein from pen, feedlot, barn, or from a Petri dish. My name is Ken Olson and I'm the Science Policy Coordinator for FAS and a member of the NCFAR board. It's my honor to be moderating our session today. Slide. NCFAR is a nonprofit, nonpartisan, consensus-based, and customer-led coalition that brings food, agriculture, nutrition, conservation, and natural resources stakeholders together with the food and agriculture research and extension community, serving as a forum and a unified voice in support of sustaining and increasing public investment at the national level in food and agriculture research and extension. Let me provide some brief background on today's topic, and our speaker will provide additional context. Lab-grown meat is a topic of increasing interest among both producers and the public. Stories appear more frequently in both popular press and trade journals. USDA recently solicited comments on labeling for meat and poultry products made using cultured cells derived from animals. This advance notice of proposed rulemaking is an important step toward ensuring the appropriate labeling of meat and poultry products made using animal cell culture technology. These actions are an indicator that these products are likely coming in the near future to a store near you. This has implications and raises questions for both producers and the public. Slide. Today's webinar would not be possible without technical support of the Council of Food and Agriculture and Resource Economics at NCFAR. I'd also like to thank the FAS Science Policy Committee for their insights in identifying our topic and speaker today, as well as their ongoing efforts to generate support for and use of science in the development of policy and regulations. Now let me introduce today's speaker. Keith Belk serves as professor and head of the Department of Animal Science, as well as an adjunct professor in the Colorado School of Public Health. Previously, he served as a professor and holder of the Ken and Myra Montford Endowed Chair in the Meat Science and with the Center for the Meat Safety and Quality Department of Animal Sciences at Colorado State University. He also served as director of the Center for Meat Safety and Quality at CSU. Now, before we start, I'd like to explain how to ask questions during the webinar to make sure you know how to engage. During the webinar, you can put questions to the speaker by typing them into the box in the control display, accessed by clicking on the question drop-down, as is shown on the slide, and sending it to the organizer. Now with that, I'll hand the webinar over to Keith Belk, who will start us off. Keith?
Thanks, Ken. Appreciate the opportunity to speak to this group today. And also appreciate the sponsorship of National NCFAR and FAS for allowing me this opportunity. Before I get started, I want to credit Dr. Mahesh Nair, who's on my faculty here at Colorado State University. I don't functionally work in research in this space at this point, but Dr. Nair does, and so he's helped me compile this presentation. And to his benefit, I think that it turned out pretty well. I'm going to spend the majority, in fact, I'm going to spend all of my time discussing the process of producing cultured products. You can skip to the next slide, please. And I feel like this audience will be fairly familiar with the biological production in the livestock industry of meat products. And so I want to talk a little bit about the process for producing cultured products and the differences perhaps that could be tied to that process relative to biology of meat production. Cell cultured product is often confused today, as are a number of technical things in the food industry, with plant-based alternative protein sources. And I think it's important and incumbent upon all of us to make sure that these two things are differentiated as consumers receive marketing messaging and social media messaging regarding their value and the science that's tied to their production. Next slide, please. If you talk about the origin for the idea of producing cultured product, you can actually go back to Winston Churchill. This is an actual quote of his that we've investigated to ensure that it's accurate. He said in 1931 that we shall escape the absurdity of growing a whole chicken in order to eat the breast of wing by growing these parts separately under a suitable medium. Essentially, it took between 1931 and 2013 to actually get to that point. Next slide, please. But by 2013, this became possible. We define cellular meat production, in this presentation at least, as meat that's made from stem cells, which tries to mimic traditional meat products. So, in other words, the growing of stem cells up and differentiating them into specific tissues in culture to mimic traditional meat production. And there's different ways that this could be labeled. It could be labeled as cultured meat, or it could be labeled as lab-grown meat. Some folks call it, you can hit the next slide, please. It could lead to a number of different linguistic terms for the product, which includes in vitro meat, artificial meat, synthetic meat. As most of you are aware, in the marketplace, a number of those that are promoting and marketing it as the next thing are marketing it as clean meat product, which has some connotation issues that could be tied to it from a safety and other perspective. Next slide, please. As I mentioned, the first cultured product that was generated was produced by Dr. Mark Post, who was at the time at Maastricht University in the Netherlands. And they did a big media event in London that probably some of you are familiar with in 2013 that allowed them to produce a hamburger that was then sampled and eaten at a news conference to demonstrate the potential viability for cultured product as a food product. Mark Post subsequently left the university and formed a new company. Next slide. Called Mosa Meat, that he now operates as one of the companies that's competing and developing the technology in this space. Since that point in time, a lot of large companies that you all would be familiar with have invested heavily in this space. The first of those that I've listed here is Cargill. Next slide, please. Tyson Foods has invested heavily in this. Next slide. The company went public to raise funding for their development of this process. Originally started as Memphis Meats. They changed their names this past May they're now called Upside Foods. But they would be one of the major players in the development of the technology towards this type of product. It was heavily funded by Bill Gates and others to develop these products. And you'll notice in this slide, they use that term clean meat. Next slide. A new product that some of you may be familiar with, it's called Just. There was some heavy media attention placed on this product last spring because it was accepted by the Singapore government as an acceptable food product. And it was being promoted in the trade media and in other media as being produced and marketed in Singapore as a chicken nugget product. The marketing behind this was a bit misleading because the actual Just cultured product is a very small proportion of an ingredient into the chicken nuggets product. And even that little amount of introduction of the cultured product into the chicken nuggets increased the cost of the product substantially, which is something we'll talk about. But nonetheless, it has entered the marketplace as a new product in Singapore. Next slide, please. And then recently, within the past 2 weeks, even some of you probably saw the news that JBS has now invested in cultured meat products with the acquisition of the Spanish company called Biotech Foods. This just occurred and closed in the last couple of weeks. And all of these efforts in sum indicate that at least at the executive levels of many of these large food companies, there's a growing interest in the potential for this product to making it to market. Next slide, please. I want to walk you first, and you can go ahead and run that slide all the way out if you would. I want to talk to you first about the process, and then I'm going to try and highlight for you some of the current technical issues that these companies are facing as they try to bring these products to market. Today, you can originate stem cells and actually do them with a biopsy, a muscle biopsy from a steer or a cow or some bovine animal. And that bovine biopsy tissue can be separated into individual cells, which you see in the upper right of the schematic, and allowed to culture in the appropriate media. That appropriate media allows those cells to rebind and grow and form myotubes, which is the actual contractile structure tied to muscle products. When that myotube is bound to a scaffolding, which you see in the center bottom of the slide, it allows that contractile tissue to actually biologically contract, which stimulates further growth of the tissue, and which ultimately allows it to develop into the cultured product that you see on the bottom left. That process in this schematic is overly simplified, and so I want to address some of the specific technical issues that scientists in this space are facing and trying to address as they move this technology along. Next slide, please. Oh, you can run that one out also, please. I want to talk a little bit about how this process works in biology. What we're harvesting with the tissue removal from the cattle is myoblast cells, which you see pictured in the upper right. And then tied with those myoblast cells is the yellow colored cell, which is actually a myosatellite cell. So a satellite cell is a cell that has yet to complete differentiation in the growth and development process to become an actual tissue. And so what you're actually harvesting that is of interest when you collect the biopsy is the myosatellite cells, which then become the formation and the foundation for the cultured growth of a new tissue in a Petri dish. Once you've harvested those cells, they begin to align and to form immature muscle fibers, which are pictured sort of in the middle of this diagram. And those immature muscle fibers ultimately grow into, with the further development of the myosatellite cells, a myotube, which is the actual contractile structure that's tied to a muscle fiber. When you allow those fibers to bind to the scaffolding, which I showed you in the previous slide, that binding, just like it would in a tendon in an animal, with continual electrical input that causes those fibers to contract, it stimulates additional growth of the myosatellites that are then bound to the myotubes, and eventually that grows into the tissue that you saw previously in the Petri dish. Next slide, please. What I want you to consider as we think about this, though, and you can run that one out, please, is the complexity of this process. What we're actually taking is stem cells or undifferentiated cells, causing them to differentiate, in the case that I just showed you, into a myoblast, which is specifically a muscle cell, and then align themselves into microtubules that consequently can begin to grow and further differentiate. But the problem in trying to stimulate those into producing a meat product in the Petri dish is what you see in the diagram at the bottom of this slide. You have to figure out ways in culture to not only produce muscle fiber cells, but you have to be able to, if you're going to mimic actual biological meat, you have to produce blood vessels and fat cells and support cells, the connective tissue that holds everything together simultaneously in the correct proportions with the correct chemical composition that ultimately allows them to become the 3D muscle tissue that you would expect to consume if it had been grown as traditional livestock. That in its complexity is a very difficult challenge. It's pretty simple today to grow individual cell lines and to have those cell lines reproduce in a Petri dish, but to have them mixed into the correct proportions and differentiated into the correct proportions that allows us to actually produce meat tissue is another matter altogether that's way more complicated. Next slide, please. So one of the first technical challenges that we have is first how to manufacture all of the different cell types that are required to produce meat. And then secondly, what kind of media are we going to grow those cells in so that they form that muscle tissue or that meat tissue, if you will. You can run that slide out also. Basically, there's different growth factors that are used at different stages of cell differentiation and growth that allows them, think of this media as food for the cells. Today in a laboratory, basically fetal calf or bovine or horse serum is added to the broth, rates of between 0.5% and 2%. And that's what's used as food to allow differentiation of those cells into myoblasts and other types of cell lines. But the precise composition of what's necessary in that serum to allow that to occur is really not well understood at this point. And in addition to that, you can purchase even today serum-free media that doesn't originate from fetal animals, but it's much less efficient. And so if a company wanted to market, for example, a true vegan product that was comprised of actual living tissue, it would probably, at this point in time at least, still require the use of products and serums from living organisms for it to be effective. Next slide, please. Next technical issue revolves around the use of antibiotics, and you can run that slide out. The common practice when cells are being cultured in solution is to add antibiotics to the solution so that those cells don't become infected with bacterial or viral contamination that they're exposed to in whatever environment that they're being grown in. And of course, you try and maintain an ultra clean environment for the culturing of those cells, but it's very difficult to prevent all contamination with viruses and bacteria. And so they add antibiotics to the serum to prevent infection that would then cause the cell culture lines to die in the plate. There's a lot of patents that have been filed for antibiotic-free mediums that can be used, but at this point, I would say the jury's still very largely out on what would be entailed in using such culture media for the propagation of these cell lines at this point, while still preventing contamination from external pathogens that can affect the cell lines. Next slide, please. You can run that one out as well. Scaffolding is the next challenge. I mentioned that scaffolding is necessary for myofibrils to grow because once you form the contractile units of muscle, they have to bind to something just like they would bind to tendons in the living organism to allow them to contract and relax. Only by contraction and relaxation will there be stimulus for those cell lines to continue to grow and reproduce in a Petri dish. And so in vivo, cells attached to the insoluble network of extracellular matrix proteins, basically tendons, through integrin receptors that are located in the sarcolemma of the muscle proteins. The sarcolemma is the cell membrane tied to these myofibers. And animal-derived or synthetic materials are available, but today the primary animal-derived product being used is a collagen scaffold, and collagen is the most abundant connective tissue in a living organism. Or they're using synthetic microbeads, which are pictured in the lower right, as the opportunity for these myofibrils to bind on each end to a scaffold that allows them then to contract and relax. This scaffolding technology is still in its infancy in terms of development, and this is something that will continue to be researched as this product continues to move towards commercialization. Next slide, please. And then lastly, well, not lastly, but the last thing I want to mention here is once all of these other previous technical challenges are able to be addressed, then you still have the issue of how do you ramp this up to production scale? And you can run that slide out. Do you do it in bioreactors? Are you able to form the mechanics to provide a continuous supply of nutrients and the other growth and media factors that are needed for cell culturing? You have to provide, obviously, a very clean supply of readily available oxygen and atmosphere. And you have to also have the capabilities of removing waste products from that are generated as the metabolism of the cell lines occurs in the reaction. And so if you think about scaling this from a Petri dish up to large production volumes like would be associated with the meat industry, that challenge becomes, at this point at least, overwhelming. Next slide, please. One of the couple final things that I wanted to mention about this is the nutritional value. You can go to the next slide. There's been some studies published about this. Basically, cellular meat does not have high levels of iron tied to it because it doesn't rely on the use of oxygen for metabolism via myoglobin in the cell culture lines. Unless you introduce the production of the protein myoglobin into the cellular matrix, there's essentially no myoglobin there. Myoglobin is essentially the halfback. If you think about the muscle fiber as the football team, the myoglobin is what carries the oxygen into the interior of muscle cells to be burnt for energy. And there just is no myoglobin present in cultured cells. And so under the low oxygen environments like Post used in his studies, you have to figure out what is that going to do to the nutritional value. And that lack of iron, among other differences in these tissues, could have a major impact on the nutritional value of the product when it's completed. Next slide, please. a lot of folks have tried to evaluate what the potential nutrient value would be of these types of products. You can run that slide out. But due to the technical challenges that are related to production, there's not any real cultured meat prototypes even available for scientists to obtain to essentially do independent sensory and nutritional assessment work on them. And so anything that you read about currently that would make reference to the nutrient value of these types of products is strictly inferred based on wherever a particular company's R&D process is at relative to addressing all of the technical issues that we've just discussed. And then the other thing that's of concern right now is cost of this product. The Mark Post product in 2013 was originally valued at about $300,000 a pound. That would obviously be greater today using that same processing technique. Companies are claiming that they've reduced that number substantially at this point. I think Memphis Meats, the last I saw of their commenting, they said that they were able to do it at approximately $15,000 a pound at this point, but that's still a very high value given the volume that they're able to generate with their current process. And then the last question mark becomes, how is this product to be regulated? You can go to the next slide. Probably most of you are familiar with the Memorandum of Understanding that was signed between USDA, FSIS, and FDA. And essentially what this allows for is the process and the mechanism and the satellite cell obtainment, I would use that word, process, is to be approved by FDA, much as a new culture line would be for any other type of product would be approved by FDA. And then the actual production process in a commercial scaled up facility would be under the auspices of USDA's Food Safety Inspection Service. Next slide, please. Some of you may ask the question, okay, where does 3D bioprinting fit into this? As you know, there's a number of efforts around the world that are focusing on this technology. I want to clearly delineate that the ability to 3D bioprint this type of product is still dependent on the ability to culture the relative mixtures of cells that are needed to actually produce a meat product. And so you still have to have all the culturing technology in place, addressing all of the issues that we've already discussed, and have those cell lines available essentially in test tubes for a bioprinter to be able to mix them in the appropriate composition and generate an actual product. This has been done at this point. This paper was just published recently this fall, and you can see that they were able to take mixtures of cell lines and produce a Wagyu beef-like product. But the dimensions of the product at this point were not commercially feasible, because it was just a tiny, tiny bit of product that was able to be generated. But nonetheless, the technology was successful. Next slide, please. And then the last thing I want to mention, and I promise this is the last thing, is the opportunity to address flavor. We all know that there's about 750 compounds at least that are involved in the development of beef flavor. Most of those compounds are developed during the cooking process. You can run that slide out. This is still a very active area of research that we don't even fully understand in biologically produced meat products. And so there's very little understanding of it so far in cultured products, none that I'm actually aware of in the refereed literature, because it's going to be dependent on the relative abundance of fatty acids and amino acids that make up whatever the meat-like compound is that is generated in the cultured technique. And so until you have a commercially feasible process in place where that is being developed, it's hard to evaluate what the composition will look like and what the consequential flavor would be for that type of product. we still don't know whether cell-based products replicate the flavor of living organism-based tissue. Next slide, please. I know you can run this all the way out. This is a very lengthy, complex slide, and I don't mean to belabor it, but I just want to show you that everywhere where there's a red circle, that is a technical issue that has to be overcome in the process flow for manufacturing a cell-based meat product that still requires additional significant science and technology. We're often asked, how long until something will be commercially ready? Well, I've already told you about a product that's being produced commercially for product in Singapore, so it is in the market. But to really be in a position to capture market share, I think to overcome these technical issues, we're probably realistically looking at maybe 10 years at least. Next slide, please. And then you have the issue of consumer acceptance, which has yet to be fully addressed. You can run that slide out. Anything that you read about in the current literature regarding the consumer acceptability of these products is merely theoretical. So you can ask consumers questions about whether or not they would be prepared to eat one of these products, but until you test a commercial product in the marketplace at the cost associated with that product, in contrast to alternative protein sources, there's no real way to measure what true consumer acceptance will be of this. We know today, for example, that consumers' greatest concern in the marketplace is genetically modified organisms. And so you have to wonder, how will they accept a product that's produced in a Petri dish versus the concept of genetically modifying other food products in the production process. Next slide, please. A paper that was just recently published addressed where consumers would be most likely to purchase this product. You can see in this results of this study, which was conducted in 2019 and just published this year, that the most likely target audience will be young people, basically the Gen Zs and millennials. Next slide, please. And then also, you can run that slide out, the two components of populations that will be most likely to be early adopters will be those that are classified by their own device as early adopters or early majority. And you can see that most of the people that are surveyed, that's about 45, 48% of the population. Next slide, please. This just gives you an idea in this study of what is important to the folks that would be considered to be early or major adopters of these types of food products. And you can see that they're always early majority components of the population. Next slide, please. And you can see that they're also very concerned about the regulatory oversight of these products and some of the labeling claims that could be tied to them. And those vary depending on where the sample was collected in the survey, but USDA, FDA approval is of most important to most of the consumers that were surveyed. Next slide, please. Last thing I want to touch on is the sustainability of this product. You can run that slide out. As you all know, sustainability and greenhouse gas emissions and profitability and efficiency is measured using life cycle assessments, at least currently. These will continue to evolve into more informative modeling systems as more research is conducted in the life cycle assessment space. But currently, the opportunity is to measure the inputs and the energy and cost that's tied to those inputs versus the outputs that are generated which includes the generation of greenhouse gas emissions and other impact areas on the environment. Next slide, please. There have been those that have tried to model what might be the life cycle assessment for cultured products. You can run the slide out. You can see that there is no commercial production yet occurring, so it's difficult to do a life cycle assessment fully. And so anything that's published in this space is essentially non-conclusive and it contains a high degree of assumption and hypothetical reasoning relative to the results of the model that is produced. The modeling should continue as the environment is a major driver for development of these products. These products are marketing towards consumers that care about particularly environmental and greenhouse gas issues. And so this will be something that this industry will need to address as these products move towards commercialization. Next slide, please. The research that has been published based on assumptions in modeling made so far are very conflicting. You can run this out. Greenhouse gas emissions from cell-based meat production are predicted to be much higher than other protein types, but there's other studies that have predicted them to be lower. I'm going to show you why that's the case. Next slide, please. This is the results of one study, and run both of those graphics out. You can see that the projected energy use for cell cultured product is much higher on average in the models that have been generated. But folks that are dealing with information about this in social media or in other forms of communication tend to present the mean or the average tied to these values. But in fact, those means and averages are of little value yet, because you see those black lines that go up and down on each bar. Those are the actual errors tied to that estimate. And you can see that the errors tied to the estimate are almost greater than the mean value itself. And so these projections at this point really have very little value. Next slide, please. There's a number of conflicting comments being marketed and promoted out there in social media and beyond. And you can run this out, please. Regarding the climate science of lab-grown products versus conventionally grown meat products. And I think many of them are misleading at this point based on our assessments in this space, because in the US at least, only 9% of total greenhouse gas emissions are really due to agricultural production, and that's across all agriculture. When you break that down into various livestock components, I think that meat production would be a relatively large component of that. Those numbers exist, but to state that you're going to completely eliminate that and have a major impact on global greenhouse gas emissions by moving to lab-grown products is a difficult claim at this point. Next slide, please. You can see in this slide that meat production that's required for major meat producing species is going to continue to increase until 2050, and that's due to the need for nutrient sustainability and nutrient security across the planet. And so that offers opportunity for production of products in this space because there's still a question mark of whether the conventional protein production sectors of the industry can even meet these demands by 2050. And so that will continue to be an issue that has to be faced on a global basis. Next slide, please. The last thing I want to mention is there is research now being publicly funded in this space. USDA, probably most of you saw, just recently this year funded a pretty large project at Tufts University to continue work on developing cultivated meat production practices. And we envision that this will likely also continue into the future with additional funding opportunities for scientists in this space. So I want to conclude by showing you just one more slide. Next slide, please. You can run this out. We question at this point whether this is actually a disruptive technology, the way it could be being promoted, mainly because of the technical issues that are still faced to ramp up a product to a commercial production scale. And we've addressed all of those that are listed there in the green. We've talked about sustainability and the effect that you could have on greenhouse gas emissions as a consequence of this practice, because agriculture in total represents less than 10% of total greenhouse gas emissions. And so then you have to think about what's the relative impact of moving in a big manner to this type of product. Nonetheless, because of the need for increased nutrition around the globe from a security perspective, there is potentially space for this if it can be produced in a cost-effective manner. We said there lastly that beef cattle provide an opportunity to use non-arable land for production of high-quality protein. And the one thing that is missed in promotion of cell cultured products is the loss of this if you were to completely shift from, for example, from beef production to beef cultured product, because you would not then capture the opportunity to harvest in non-arable, non-agriculturally producing areas, the grasses that are available, like we produce here in Colorado in our mountains. And so that's something that must be considered as this moves forward. And then lastly, in those processes, beef cattle are able to upcycle byproducts and reduce and produce much lower impactful mechanisms of greenhouse gases, particularly methane. And so that will also be something that will have to be considered as these cultured products find their place in the overall marketing scheme. With that, I want to stop, and you can switch to the next slide, and we'll open it up to any questions that you might have. Thanks again for the opportunity to share this with you this morning.